Every year, humans eat over 70 billion chickens, and it's no surprise. They're lean, protein-rich, and packed with vitamins and minerals. But how did we build the bird behind your McChicken? To find out, we need to go back 7,000 years to Southeast Asia. Here we find the flamboyant wild ancestor of all chickens, the red junglefowl. These little fellas were the first bird or reptile to have their whole genome sequenced. Coming in at 23,000 genes and roughly 1 billion base pairs, the junglefowl's genome was 1/3 the size of all previously sequenced mammals, and presented some unique features. Its chromosomes came in 2 different sizes, large and small, and unlike in humans, the female was the one with 2 different sex chromosomes, making her heterogametic. The genome has since become an invaluable reference point, helping us follow the genetics of today's oversized meat monsters. But that leaves the question, why did we start breeding this bird in the first place? Well, it boils down to the junglefowl's unique breeding strategy. In this part of Asia, the dominant bamboo species bloom just once every 50 years, releasing a flood of seeds. When this rare feast arrived, the junglefowl's reproductive system went into overdrive, with hens laying eggs by the dozen. Humans saw this and realized we had the supplies to stuff them with more seed than a bamboo forest ever could, ultimately producing round-the-clock omelette machines. By breeding the most prolific hens, we transformed the junglefowl into the layer. Comparing to the junglefowl's genome, almost all layer species have genes that have undergone a selective sweep. Layers share mutations in a gene for TSHR. TSHR signaling in the brain regulates day-length-based reproductive cycles, explaining why these birds don't breed seasonally. But layers were just the first step. Quickly, we realized hens didn't just lay protein, they were it. Breeders spent the following years chucking their girthiest cock and hen in a 1x1 and hoping for the best. But with the end of World War II and a boom of hungry mouths to feed, progress was far too slow. We needed meat more than ever before. Eventually, the USDA was done leaving things to chance. They announced the Chicken of Tomorrow contest, a nationwide initiative that aimed to produce a chicken with more white meat than a turkey. Breeders submitted over 2,000 of their very best birds, which were evaluated meticulously on 18 traits. So who was this peak poultry? I present the Arboracre breed, a bird with rapid reproduction, adaptability, and efficient feed conversion. By 1964, the company behind the Arboracre was pumping out chickens quicker and cheaper than anybody else. At the cost of diversity, these titans conquered the world's dinner plates. In their takeover, Arboracres rapidly spread genetic mutations that granted them their physique. As a result, almost all broilers now share specific selective sweeps in regions linked to muscle growth and metabolic control. Notably, a selective sweep at the TBC1D1 locus, right at the spot that drives the rapid growth of broiler chickens, might be the leading explanation for their terrifying girth. Human mutations in this gene have been linked to obesity, but given these birds are destined for a gym bro's dining plate, that's only a positive. By selectively breeding only the best mutant hens, we built layers that produce 25 eggs a month and broilers that bulk up to 2 kilograms in less than 35 days, a speed twice as fast as the 1978 junglefowl. Chicken is now both the cheapest and most environmentally conscious farmed meat we have. By cracking open the junglefowl's genome, we uncovered the blueprint to producing drumstick dispensers on steroids.